Hello, good day. So let's start with another new topic, more on the skeletal system. Okay, the skeletal system is the second system after the integument system. The significance of this system is considered as the most important organ system in the study of the vertebrate morphology. The skeleton shows the broad outlines and evolutionary trends of vertebrate phylogeny. It also provides reliable information about the specific adaptation of vertebrates such as posture and locomotor adaptations. It tells us about other organ system. Why? Because it also directs us where the bones are located and the muscles is interacting. So the origins and insertions of most skeletal muscles are on the bones, and they have leave a mark showing its position and extent of contraction. The foramina found in our bones will reveal the size and the course of the cranial nerves. The brain case may reveal the relative development of the different parts of the brain. The nasal chambers, orbits, or other spaces, or the otic cavities give information about the sense organs. Even some blood vessels leave traces on the skeleton. Due to its hardness and durability, skeleton becomes the fossilized and the study of past vertebrate life in most bases on fossils. So what are the main functions and general organization of the skeletal system? Actually, there are many functions of the skeleton. This includes give shape to the body and supports its weight. Like we have the vertebra and our long bones in the inferior part, our legs, which supports and receives all the weight of the body. Offers also a system of levers that aids muscle to produce contraction. Our arms are examples of a lever. It also protects soft parts such as nerve, blood vessels, brain, and other viscera. So one good example, you have there the brain enclosed by a skull, and the heart and the lungs are protected by the ribs. As a protective and supportive system, the skeleton can be divided into two types. So, we have here the exoskeleton, the protective structure on the outside body of the animal. It was formed from within the integument, and epidermis give rise to keratin, and the dermis give rise to bones. The second one, we have the endoskeleton, the protective structure of the inside of the body, formed deep within the body from mesoderm and other sources, not directly from the integument. But in in the animal kingdom, actually we have the third type. We have the hydrostatic skeletal system. Mostly found lang ito sa mga invertebrates, lower invertebrates like the sponges, cnidarians, and annelids. So, hydrostatic skeletal system. This was made up of water, and the water exerted a pressure that gives shape to the animal. Exoskeletal system usually found sa mga higher invertebrates like what we have the echinoderms, the group of sea stars, urchins, the insects, mga arthropods, and also what else? Arthropods. Examples of arthropods, we have the crustaceans, mga insects, mga myriapods. Those are example of under the phylum Arthropoda. And mga endoskeleton, usually found sa mga higher vertebrate groups, started from the fishes going to the class Mammalia. So we have two separate components of the skeletal system. We have the the cranial skeleton, or represented by the skull. And we have the postcranial skeleton, which includes the axial and appendicular skeleton. The axial skeleton includes the notochord and the vertebral column, the ribs, and the sternum. While in the appendicular skeleton, so this is the appendicular, includes the skeleton of the median fins of the fishes, pectoral and pelvic girdles, like what we have here, the humans, and the skeleton of the paired fins and limbs. So our limbs, our hands, and our feet belong to appendicular skeletal system. So this was composed of mineralized connective tissue, like we had the dentine, enamel, the cartilage, and mostly the bony tissue. So the skeleton developed from mesenchyme, differentiated into scleroblast, which give rise to osteoblast, then the osteoblast to odontoblast, chondroblast, and ameloblast. Well, odontoblast, this type of cell, produce dentine. So this one gives, usually found sa ating teeth, that produces a hard covering of our tooth. Ameloblast produce enamel. Chondroblast produce cartilage, and osteoblast produce the bone. So give rise to osteocytes, or we call it the bone cell. So let's proceed sa cartilage. Okay, cartilage forms an important part of the endoskeleton in all vertebrates. So the cartilage is a vascular tissue. The development begins with a blastema, an aggregation of embryonic mesenchymal cells differentiates into tissues such as chondrocytes, or we call it the cartilage cells, or osteoblasts, bone-forming cells. Now, the cartilage characteristics, aside from being avascular, when we say avascular, they don't have blood vessels. Chondrocytes receive nutrients and excrete waste by simple diffusion through the matrix. Hyaline cartilage is the most abundant cartilage. So this is hyaline cartilage. That is the most abundant cartilage tissue. It is homogeneous and translucent. So based in microscopic sample, they are very clear. It depends on the dye. If you use yung blue or yung pink dye, it is homogeneous and translucent. Found in distal parts of the ribs, or the costal cartilage, the ends of the ribs, the articular surfaces of long bones, and most of the cartilage of the larynx, trachea, and bronchi. So these are the organs where the hyaline cartilage is located. Other types include the elastic cartilage. You have seen here the middle portion, the elastic cartilage tissue. Usually found in the external ear. That's why our pinna, or we call it the external ear, is kind of flexible. Epiglottis. So these are some of the areas where the elastic cartilage is located. Because added structure of the elastic cartilage, which cannot be found in the hyaline, elastic cartilage have elastin. So what makes darker appearance in the matrix, meron silang elastic fibers. Next type, we have the fibrous cartilage in the intervertebral disc and attachment of tendons and ligaments. So the fibrocartilage. The fibrocartilage is an ideal cartilage that receives shock or absorb weight, support also. So let's proceed to another tissue. Ah, in this diagram, you have here yung mga fishes that mostly made up of cartilage skeletal system. So we have the class Chondrichthyes represented by the hagfish, the lamprey, and nastosome. When we say nastosome, they have already the lower jaw. So this group could be the shark, mga hammerheads. Those are examples of the members of the shark family. So let's proceed the bones. The bones comprise most of the endoskeleton of higher vertebrates, started from the bony fishes, or the osteichthyes. Development begins with a blastema, an aggregation of embryonic mesenchymal cells differentiates into tissues such as muscle, cartilage, and bone. So these cells develop into either fibroblasts. So ang mesenchymal cells develop to different types of cells. So aside from being a muscle, a cartilage, it develops to a bone. So it will develop further into fibroblast or osteoblast. Then fibroblasts form collagen and osteoblasts form bone cell called the osteocytes. Then the third one, the matrix, quickly become impregnated with calcium and other organic salts which make it up to 30% of the bony tissue. So sa bony tissue, that's why it is compact. The matrix is very solid compared to hyaline because it was impregnated already with other minerals like the calcium and some inorganic salts. So each bone cell lies in a lacuna. So we have here sa this is the lacuna. So we have the macro visual of the bone tissue. Inside the lacuna, we have here the osteocytes. And at the center, we have the Haversian canal or the central canal. This is the area where the blood vessels, the nerves present in the bony tissue. Since they cannot penetrate the matrix, so this is one way, a certain adaptation where the other structure could deliver the nutrients needed by the osteocytes. So another important part, we have here the canaliculi. Okay, the osteocytes trap in the lacuna, diba? So it may gain access to the tissue fluids through the tiny canals. So these tiny canals called canaliculi. So the canaliculi penetrate the lamellae. So this is one way to distribute the nutrients going to the osteocytes. Okay. So the organic component is primarily made up of collagen, which give rise bone great tensile strength. The function, it gives support and movement via attachment of soft tissue. So protects also the vital organs. It is also the major site of red marrow or for production of blood cells. And it also plays a role in metabolism of minerals such as calcium and phosphorus. There are two basic structural types of bone. We have here the spongy, or call it the cancellous bone, and the compact bone. Okay, compact bone forms the outer shell of all bones and also the shaft of the long bones. So that's why it is a good structural support for the organism. That's why we could walk, we could stand the weight, could support the weight of the body. The spongy bone naman, this is the spacious part, is found at the expanded heads of long bones. So the expanded heads of long bones. So if you're going to check the diagram, so this is a spongy area of the bone. So this one, the shaft and portion of the heads are made up of compact bone. And what you can see here, the red tiny mark at near the ends of the long bones, we have there the spongy bone. So bones are tightly surrounded by periosteum. So this is the periosteum, another covering, a dense layer of connective tissue. So the growth of bone is by apposition, since interstitial growth is inhibited by the presence of the rigid matrix. So bones are formed in two ways: either intramembranous ossification and the endochondral ossification. So the bones, there are two ways where it is developed: the intramembranous ossification, a process by which membrane bones are formed, such as bones of the lower jaw. We have the skull and the pectoral girdle, the dentine, and other bones that develops in the skin. So in some vertebrates. So example, we have the telos, urodel, and the apodans. While ang endochondral ossification, process by which bone is deposited in pre-existing cartilage, forming a cartilage bone or a replacement bone. So meaning it derived first from a cartilage cell when there's deposits already of minerals and salts, calcium. The cartilage cells hardens, especially the matrix, then it transforms into a osseous bone, so that, or a replacement bone. So let's proceed sa cranial skeleton. Okay, the skeletal framework of the head is the skull, composed of the following main components. We have here the main case, the brain case, the cranium or the neurocranium, which surrounds and protects the brain. So this one. The sense capsules, associated in development with the olfactory, auditory, and visual sense organs. So we have here this the capsules, and in this side, some temporal region for the auditory, and numerous components of the visceral skeleton. So it is derived from three different embryonic components. So the cranial skeleton derive or develop from three embryonic components. We have here the chondrocranium, composed of cartilage, which contributes to the base of the skull, includes the sense capsule, and most vertebrates is replaced by bone. So from cartilage, it replaced into bone. Next, we have the dermatocranium, made up of membrane bones, which the roof over the chondrocranium. So this is the roof. So this is a dermatocranium. So that frontal bone, temporal, and a part of occipital will made up as the roof of the brain. And the splanchnocrinium, derived from the visceral skeleton, is primitively cartilaginous. So it says that the splanchnocrinium derived from the cartilage. Now let's discuss the chondrocranium ossification centers. So chondrocranium, primarily the brain case. So this is the brain case. Surrounds the brain and special sense organs like the eyes, yung auditory, dito sa ang auditory. And since we don't have the frontal skull, yung the capsule where the eyes is encased. So next, develops from the paired cartilages, most of which eventually fused with one another. So it is derived from the cartilage. So development of the chondrocranium. So after the formation of the embryonic brain, the mesenchyme differentiate to form a membranous layer called membranous cranium. So later on, when the chondrocranium develops further, it has its own ossification centers. So we have the occipital center, the sphenoid center, the ethmoid center, and the otic center. Again, sa other books, we have here, so occipital center, the sphenoid center, the ethmoid center, and the otic center. So let's have the occipital center. Now the occipital center, the cartilage surrounds the foramen magnum. So this is the foramen magnum. May be replaced by as the main four bones. One basio-occipital. So this is one basio-occipital. Two exo-occipital on the lateral part. And one supra-occipital. And we have the last one, the interparietal bone, which composed of two areas. Next, the sphenoid center. Okay, the sphenoid center form the basisphenoid bone and the presphenoid bone, and the side wall above basisphenoid and presphenoid form into orbitosphenoid, plurosphenoid, and alisphenoid. So the sphenoid center, so this is the sphenoid center that derives from the cartilage, will develop the basisphenoid, the presphenoid, the orbitosphenoid, the plurosphenoid, and alisphenoid. So ito yung portion, the sphenoid center. Next center, we have the ethmoid center. So this is the ethmoid center, a small portion of it. Tend to remain cartilaginous and form cribriform plate of ethmoid and several ethmoturbinal bones. So this is very small, and followed by the otic centers. The otic centers, the cartilaginous otic capsule is replaced by lower vertebrates by several bones, like what we have the prootic, opistotic, and epiotic. So these are the three bones, the prootic, opitotic, and epiotic. Okay. So we have different types of skull. So each classes were also classified, and this could be used to classify the vertebrate groups based on the type of skull. So first type. So types of skull based on fenestrae. So temporal openings. Asa ang temporal part? Sa temporal part is located sa lateral side ng skull. So above your ear, if you are going to touch your temporal part, that is above your ear or external ear. So that's the temporal part. So temporal openings. So we have here the anapsid. When we say this is a primitive type of skull that has no temporal fenestrae, possessed by turtles and other primitive reptiles. So this is the anapsid. So you can see here we have the optic foramen, no temporal opening. Diapsid skull. The diapsid skull has two temporal fenestrae, possessed by most members of Diapsida, including crocodiles, birds, and lizards. So we have 1 and 2. Di means 2. So that is a diapsid skull. So this is for the eye, optic foramen. So the temporal side have 2 foramen. Next, Europsid skull. This is derived from diapsid, the skull where the lower temporal fenestra is lost. So, if you're going to compare with the diapsid, you have the upper opening and the lower opening. In the Europsid skull, the lower temporal fenestra is lost. Next, we have the synapsid skull. Synapsid skull has no fenestra located in different place than the Europsid skull. So this is the diapsid skull. Okay, if you're going to check on the other diagram, so we have here the diapsid skull, another diapsid. So you have there the upper and lower temporal opening, the anapsid. So the anapsid, the synapsid, and anapsid, another anapsid. Now let's proceed sa dermatocranium. Dermatocranium composed of dermal bones that overlie the chondrocranium and splanchocranium. So, by the diagram, we have here the neurocranium, and this is the dermatocranium. So that's the position of the dermatocranium. It completes the protective cover of the brain and the jaw. So this is our lower jaw, lower mandible sa atin in humans, and we have the brain case. What are the parts of the dermatocranium? Okay. Modern fishes and amphibians have simple skull, and the number of dermal bones present is reduced. Some have tended to be lost or fused. So in amniotes, dermal bones predominate, forming most of the brain case like us. And the lower jaw, so we have lower mandible. They are divided into 6 series bones. So another way, you have to memorize what are the 6 series bones of the dermatocranium. Okay, we have the facial series. So we have the facial series. So sa human, we have the facial bones comprising the frontal bone, the parietal bone, the sphenoid temporal bone, the sphenoid bone, the smaller portion, the nasal bone, the zygomatic bone, the vomer bone that divides your nasal passage, the maxilla, we have 2 maxillar bone, and the mandible. So these are the facial series. So encircles the external nares forming the snout. Orbital series encircles the eye, defining the orbit. So what are the bones also? The frontal bone includes also the nasal, the maxilla. So those are the bones that comprises the orbit. Temporal series lies behind the orbit, completing the posterior wall or the brain case. So again, the temporal series, it lies behind the orbit, completing the posterior wall of the brain case. So these are the squamosal or the quadratojugal completes the series from the cheek. So that is this is the inner bone. Fenestrae openings of the outer brain case make of many tetrapods arise with the region in association with the jaw musculature. Next, we have the vault series or the roofing bones. Located across the top of the skull, covering the brain beneath. So this will serve as the floor of the brain. So this includes the frontal anteriorly and postparietal posteriorly. Next, we have the palatal bone or the palatal series. Dermal bones of the primary palate. So if you're going to open your mouth, the roof of your mouth that will serve as the floor of the other part. So dermal bones of the primary palate covering the roof of the mouth. This includes the largest and the most medial pterygoid, the vomer, the palatine, and the ectopterygoid. Sa fishes naman, we have the unpaired medial bone. Parasphenoid is also present. Next, we have the mandibular series. Encases the Merkel's cartilage. This includes the tooth-bearing dentary and one tooth splenial on the walls in your lower mandible. The angular posterior corner of the mandible. So this is the angular posterior corner of the mandible. The left and right mandible usually meet anteriorly at the midline called mandibular symphysis. So the left and right. That uniting them into an arc unit. So when it is united, you have there the arc already. Okay. So those are the parts of the matocranium. Again, we have the facial series, the orbital series, the temporal series, the vault series, and the palatal series. And the mandibular series, before we had to forget. Next, we have the splanchnoscranium. Okay, splanchnoscranium. Okay, the splanchnoscranium, this is the ancient corded structure associated with the filter feeding surfaces. So, makes up the skeleton of the jaws and gills in fishes and amphibians. The may remain cartilaginous in some fishes or become covered by dermal bones. It arises from the neural crest cells departed from the sides of the neural tube and migrate into the walls of the pharynx between successive pharyngeal slits. So, this is the fate of the splanchnoscranium. So, we have there the Merkel's cartilage, palatoquadrate, rostrum, labial cartilage, basial cartilage, ceratohial cartilage, hyomandibular cartilage. So there are 13 that is associated to splanchnoscranium. Okay, next. Now, the skull also was, aside from types of fenestra, another character to distinguish other vertebrates by the types of jaw attachment. So, jaw attachment is another character to classify vertebrate groups. So, we have the paleostylic characteristics of agnatans. So, mostly sa fishes with no lower jaw. None of the arches attach directly to the skull. Euautostylic, the earliest jaw condition, found in placoderms and acanthodians. The mandibular arc is suspended from the skull by itself without aid of the hyoid arc. So, there's no hyoid arc. Amphistylic. Okay, amphistylic found in early sharks, some ossixians and crossopterygians attach the brain case through primary articulations, arterially by ligament connecting to palatoquadrate to the skull, posteriorly by hyomandibula. So, the red portion is the hyoid mandibula. So, the green portion, this is the chondrocranium. And the yellow one is the mandibular derivatives. And its arc. What about the hyostylic? Hyostylic found in most modern bony fishes, like tilapia. The mandibular arc is attached to the brain case, primarily through the hyomandibula with the aid of symplectic bone. Fifth, we have the metaostelik, found in most amphibians, reptiles, and birds. The mandibula is attached to the brain case directly through the quadrate bone, formed in the posterior part of palatoquadrate. Next, we have the sixth one. We have the craniostelic, found in mammals. So the entire upper jaw is part of the brain case, but the lower jaw, called dentary bone, suspended from the dermal squamosal bone to the brain case. So craniostelic, found in mammals. So since we are mammals, we have this type of attachment. Okay, next. So that's the cranial skeletal system. So we're done on the first part of the skeletal system. Now let's proceed on the second part of the skeletal system.